Assalamu alaikum everyone. I'm Zaina and I'm a third year medical student, and I'll be covering this session on biliary secretion and enterohepatic circulation. Okay, so the way I did this, before we begin, the way I organize this lecture is a little bit different than how your doctor did it. I'll start off by talking about synthesis of bile acids, and then I'll go on to talk about changes that happen in bile acids, how it's stored, when it's secreted, and then we'll talk about the effect that it has on the hydrolysis and absorption of lipids. Lastly, we'll talk about how it gets reabsorbed and reused, and then we'll end with the clinical relevance of bile acids. Okay, so before we get into the physiology of synthesis and secretion of bile, let's look a little bit into the anatomy. So we have two hepatocytes here, right? Between the two hepatocytes, we have something called a canaliculi. Hepatocytes are basically cells of the liver that synthesize bile acids, and then they secrete it into the canaliculus. And once the bile is secreted into there, bile canaliculi, they combine to form intralobular ducts. So these here, as you can see, are intralobular ducts. These come together eventually to form canals of Hering. So these canals here are called canals of Hering that drain into hepatic ducts, intrahepatic ducts. And then later we'll see where bile goes from that. Okay. Now let's begin with talking about the synthesis of bile acids. Okay. Now it all starts with cholesterol. Cholesterol, it undergoes 7-alpha hydroxylation to form cholic acid and chenodeoxycholic acid. These are primary bile acids. Now, I don't really need to know the names of this, but the most important part you need to know on this slide is the rate limiting step, that's 7-alpha hydroxylation. Okay. Um, so, okay, so once we have our primary bile acids, these get conjugated. What do you mean by conjugated? We have addition of glycine and taurine. These amino acids that get attached to the choline and chenodeoxycholine to form glycocholic acid and taurochenodeoxyglycolic acid. Again, we do not need to remember these names. Now we need to know that primary bile acids, they get conjugated. This happens by addition of glycine and taurine. Okay, now why do we do this? Now, conjugated bile acids are basically more water soluble. So we do this whole thing to make it more water soluble. Okay, now once we have synthesis of bile acids in the hepatocytes, we have to secrete it into the canaliculus, right? Now, like I said, we have synthesis of bile acids in hepatocytes, but we also have some percentage of bile acid that comes from the circulation. Now, once you have bile acids getting secreted into the canaliculus, it goes through the biliary system and then gets secreted into the intestine. It does its effect, and then it gets reabsorbed to go back to the liver. Once it gets reabsorbed, it has to get resecreted, right? So it enters the cell through two proteins, OATP, that's organic anion transporting protein. This brings in unconjugated bile acid. And then we have NTCP, that's sodium taurocholate co-transporting peptide. This brings in conjugated bile acid. Okay, the unconjugated one, it gets converted to conjugated, and conjugated bile acid, it gets secreted into the canaliculus through this very important protein that's called bile salt explode protein. Okay, that's ATP dependent. So this protein here is very high yield. If there's something going to get asked about in this slide is this. Okay. Now moving on, now that we have our bile acids getting secreted into the canaliculus, there are certain changes that will happen to it. Okay. Now in the bile ducts, we have cells called cholangiocytes. These make the bile more dilute and more alkaline. How does it do that? And we have this hormone called secretin. Okay. Now once secretin is released, it binds to receptors on the cholangiocytes and it increases the concentration of CAMP inside the cell. Okay. Once it increases concentration of CAMP, it activates certain receptors called CFTR receptors that pump chloride ions outside the cell. Okay. Now after this happened, the next step is to bring back to bring back the chloride inside the cell. This happens through anion exchangers called chlorine bicarb anion exchange. Okay, now chloride comes back into the cell and bicarb is in the ductal lumen. Now we know bicarb is what makes the bile more alkaline. So the presence of bicarb here is what makes it more alkaline. Now, how do we get chloride and bicarb? Now we know that we need chloride to pump it out of the cell, right? And we also have bicarb that gets effluxed out. So how do we get the concentration of chlorine and bicarb inside the cell? We have two transporter proteins. One of them is sodium potassium 2-chloride channels that exists on the basolateral membrane. It's this part that faces the blood. These channels, they bring in chloride inside of the cell. And then the next protein transporter is sodium bicarb cotransporter. This brings bicarb inside the cell. Okay. So now that this whole thing is basically to make the bile in the duct more alkaline, right? We also said that we need it to be more dilute. How does that happen? Now we have movement of sodium that moves from the interstitium into the duct and paracellularly through tight junctions. And once that happens, water follows. So if water follows, the water always makes something more dilute. So the bile is now more alkaline and more dilute. Okay. This is basically everything I said in words. You guys want to read it? Okay. So now that we have our bile acids secreted, and we don't need bile acids to get secreted continuously, right? So we have to store it somewhere. Where does that happen? In the gallbladder. Okay. So once we have hepatocytes secreting bile into the bile ducts, the enterohepatic bile ducts, they combine to form the common hepatic bile duct, right? And because we don't need bile here, this is basically in times where between meals, okay? So where you don't have food, so you don't need bile acid to get secreted. So the sphincter of Oddi here at the end would be closed. And bile, once it gets released, and it can get released through the sphincter into the intestine. So there's this pressure here that builds up and it goes back to it goes to the gallbladder, okay? And that's where it accumulates. You can see the gallbladder has expanded because you have bile acid that gets stored. And yeah. Okay. Now while it's in the gallbladder, there's certain changes that happen to the bile. Okay. Now gallbladder, it tries to make the bile more concentrated. How does it do that? Now we said that if you want something more concentrated, you remove water from it, right? And when you remove ions from a lumen, the water would follow, and that would make it more concentrated. So let's look into how that happens. We have certain proteins here on the surface. They're called sodium hydrogen exchangers. They bring sodium inside the cell and they pump hydrogen out. Okay. Similarly, we have other transporters that bring chloride into the cell and bicarb outside. So sodium chloride, these both move through the basolateral membrane into the interstitium. Okay. Now, when that happens, because sodium chloride they got pulled away from the ductal lumen, water also follows. It follows through these channels called aquaporin channels. Okay. So now we have our bile that's more concentrated. Now, like I said, we have hydrogen and bicarb that gets released into the duct, right? What happens to these two? They combine through an enzyme called carbonic anhydrase to form water and CO2. Now, water here, it moves paracellularly and it gets absorbed. Okay. Another thing that happens here is that the gallbladder, it makes the bile more acidic. How does it do that? It's because these proteins, sodium hydrogen exchangers, they are present in a much larger number than chloride and bicarb exchangers. So that means you'd have a higher concentration of bicarb in the lumen. Sorry, you'd have a higher concentration of hydrogen ions. This makes bile more acidic. Now you might wonder why we're making more acidic, because in a few slides ago I said that cholangiocytes, they make it more alkaline, right? So how does it make it more acidic here? So basically the whole point of the gallbladder doing all this is to make the bile more concentrated. We don't really care about the pH. Now we need bile to be alkaline because when when it goes through the stomach and into the intestine, it's basically acidic. So we need something to neutralize it. And this is why in cholangiocytes, they make bile more alkaline, okay? But here, gallbladder, when it stores it, it temporarily makes the bile more acid. But eventually, when it gets released into the intestine, it combines with pancreatic secretion that make it more alkaline and neutralize the chyme, okay? Okay, now let's look into differences between liver bile, and we said The hepatocytes, they secrete bile acid. And then we also have gallbladder bile that makes it more concentrated, right? Because it makes it more concentrated, you can see here that you have less water in gallbladder bile and more bile salts. This means gallbladder bile is more concentrated. Similarly, now we said here that sodium and chloride gets absorbed. So you'd find the concentration of sodium and chloride would be much less. The amount would be much less in gallbladder than the liver. Okay. So now that we have the gallbladder storing bile, when do we need to secrete it? Now, when you have a meal, you need bile, right? To hydrolyze lipid, to absorb lipids. So when you have a meal, gallbladder, it starts contracting. It releases the bile, and that which passes through the common bile duct, and then the sphincter of OD opens because, it opens and secretes bile into the intestines. Okay. Now let's look into more details of how this happens. Now, when you have a meal with lipids and proteins, they get digested in the stomach and they go through the duodenum, right? In the duodenum, we have certain cells. They're called eye cells, okay? So these food digested products, they stimulate these cells to release something called cholecystokinin, okay? And cholecystokinin, it goes to two pathways. One, it goes to the gallbladder, it contracts it, releases bile, and secretes it into the duodenum. Okay. Another pathway, cholecystokinin, it activates vagal afferent neurons that go to a part in the brain called dorsal vagal complex. Okay. It sends out two afferent neurons. One that goes to the gallbladder, it releases acetylcholine, contracts it, and again it releases bile and secretes it. And the other pathway, that is the neurons, they go to sphincter of OD, they release VIP and nitric oxide and cause relaxation of the sphincter to secrete the bile. Okay. This is everything that I said in words. Okay. Now, once we have bile getting secreted, we know they act on lipids, they break it down and they absorb it, right? Let's see how that happened. Okay, so we have two types of lipids, long-chain dietary lipids, long-chain triglycerides and medium-chain triglycerides. The long chain is the more important one here because it's more available. Okay, so the two things you need to hydrolyze lipid, lipases and bile salts. We have two types of lipases, gastric lipases and pancreatic lipases. Pancreatic ones are the more important ones. They work much more on lipids. Okay. So bile salts, this is basically what it looks like. You have this green part are hydrophilic and the yellow part is hydrophobic, okay? And they do two things, emulsification and solubilization. Let's see what emulsification is. Now you have this fat globule that you get from your food. The bile salts, they act on it and they make it into smaller droplets, okay? And then they attach themselves on the surface like this. Now you have bile salts on the outside and the lipid on the inside. This whole structure is called a micelle, okay? It stabilizes the smaller droplets. Now, why are we doing this? Why are we making the smaller droplets? Because we're increasing the surface area for pancreatic lipases to act on it. Okay? And once they do, they would break it down into free fatty acids and two monoglycerides. Okay. So this is how it looks like. And you have bile salts on the outside, and the lipase, it acts on the lipid on the inside to form lipid breakdown, free fatty acids and two monoglycerides. And this is called the mixed micelle because you have the mixture of lipid breakdown. It's called mixed micelle. Okay? And this makes it more soluble. This whole structure makes it more soluble. Okay, we have something called a critical micellar concentration. And I see here you have one bile salt. One of it cannot make up a micelle, right? You have two, you still can't make up a micelle. And in this example, you can see you have six bile salts together, they can make a micelle. And this concept, the doctor didn't really say it. This is just helpful to know. Okay. Now let's look more into details of how pancreatic enzymes, they work. Okay, so this is a micelle, and we have lipid on the inside, and we need to break it down, right? So lipase comes, it's because we have bile salts here, it cannot reach the lipid and break it down. So instead, we have something called colipase. This colipase, it comes and binds to the lipase, and it allows it to anchor to the lipid and break it down. Okay. Another thing that does is that it lowers the optimum pH of the lipase from 8 to 6. And you know, inside the intestine, we have a pH of around 6 acidic. So it's so the lipase would work more. Okay. Now, once we have the lipid broken down, we reabsorb it to the intestines, right? Now, coming back to the structure of a micelle, we have bile salts on the outside, and then we have the lipid breakdown on the inside, okay? So this micelle, basically, it acts as a shuttle for these lipid breakdowns to get absorbed into the enterocytes. This happens by diffusion. So once they're inside, these lipid breakdowns, they combine with free fatty acids to reform their lipid molecules. Okay, so monoglycerides, it combines with free fatty acid, and it forms triglycerides. Okay, so once you have these lipid molecules, they combine together, and this process of recombining and forming lipid molecules, it happens in the smooth endoplasmic reticulum. Okay, now once you have these lipid molecules, they combine together to form this structure called a chylomicron. And this happens with the help of an apolipoprotein known as ApoB48. Okay, so these lipid molecules, they come together with ApoB and they form a chylomicron. This whole process, it happens in the rough endoplasmic reticulum. Okay, the purpose of this chylomicron is basically to make the lipids more hydrophilic so that they can go into the circulation. Okay, before we get into reabsorption, does anyone have any questions? Guys, let me know if I'm going too fast. The last slide, this one? This one? Okay. Okay, so you know how we said the micelle is formed, right? We have bile salts on the outside, and then we have the lipid products on the inside, right? Now this whole structure, it acts as a shuttle, and it allows these lipid molecules to diffuse into the enterocyte. Okay? Now once they're inside the enterocyte, they combine with free fatty acids to again form the original lipid molecule. Okay, so monoglycerides. Now we have monoglycerides, the breakdown of triglycerides, right? And when they go inside the cell, this is basically forms for it to go inside the cell. Once they're inside the cell, they combine with free fatty acids and they form the triglycerides. Okay. Okay, and these triglycerides and all these lipid molecules, they combine together. Now we need them to be in the circulation, right? Now because they're hydrophobic, they can't get into the circulation. We need them to be more soluble, right? So for that, they combine into this structure. This is called the chylomicron. Now how do they combine and they become hydrophilic like that? By the help of this protein called ApoB48. Okay, so ApoB48 comes in, it gets them all together and it forms this structure. This is known as the chylomicron. Okay, and then it gets excitosed into the blood and it gets circulated. Is it clear? Okay. Now, once bile, so this whole thing, it happens in the intestines, right? The duodenum. The bile acts on the lipids, it breaks it down, and causes it to get reabsorbed. So once bile is done with this function, what happens to it? It gets reabsorbed through the portal circulation back into the liver, okay? And in the portal, it's bind to albumin. Now, this whole thing, the reabsorption happens in distal ileum, okay? So we have two types of bile acids, conjugated and unconjugated ones, okay? So both of them, they get reabsorbed. But each of them, they have their own way of getting reabsorbed. Okay. Now let's look into the absorption of conjugated bile acid. This is very simple. They enter into the cells in the ileum through a transporter called apical sodium bile acid transporter. Okay. Once they're inside the cell, they go into the portal circulation through another one called organic solute transporter. So all you need to know from here are these two transporters. Okay. Okay. Now I said that we also have deconjugated bile acid, right? Now this deconjugation basically happens in the distal ileum, and it's done by ileum bacteria. So bacteria, it deconjugates the conjugated bile acid, like cholic acid and deoxycholic acid, into into deoxycholic acid and lactic acid. Now, again, we do not need to know these names. We just need to know that the ileum bacteria in the distal ileum, they deconjugate and dehydroxylate it to make it more lipid soluble and less water soluble. And why do we need it to be more lipid soluble? Again, so it can get reabsorbed into the circulation. Okay, and that happens by passive diffusion. Okay, now let's come back to the slide that we discussed in the beginning. Now, I said how some percentage is made in the hepatocytes and some comes back to the hepatocytes through what we just said, the enterohepatic circulation, right? When it comes back, it enters hepatocytes again through these two transporters, OATP and NTCP. And like I said, the conjugated ones, it gets resecreted into the canaliculus through bile salt export protein. Okay. Now, this table is basically a summary of everything we just said. We have, one sec. Okay. Okay, now let's look into it based on the phases of digestion. The first one we have a cephalic phase. It's basically when you eat food, the taste of the food, this acts as a stimulus to cause VIP and nitric oxide to get released through the vagus nerve. Now, does anyone remember what these neurotransmitters do? Yes, yes, okay. Causes gallbladder contraction and relaxes sphincter of Oddi. Okay, this would cause it to get secreted, bile to get secreted. Okay, next, the gastric phase. When food reaches the stomach, we have gastric distension. This acts as a stimulus. Again, it releases the same nitric oxide and VIP, and it causes the same effects. Okay, in the intestines, again, we said that we have fat digestion. Fat digestion products, they stimulate certain cells in the duodenum. Does anyone remember what those cells are? They're eye cells, okay? They release something. What is that? CCK, exactly. Okay. So once CCK gets released, we know it does the same thing. It causes gallbladder contraction and relaxes the sphincter. Okay. Now, lastly, in the intestine, we have a stimulus that's acid in the duodenum. When it's acidic, we want to make bile more alkaline, right? So that it can neutralize the acid in the duodenum when it gets secreted. So what makes the bile more alkaline? There's something that the hormone that gets released that attaches to the cholangiocytes and causes and makes it more alkaline. Yeah, the secretin. Okay. Okay. Now, lastly, when we have bile acids and they get reabsorbed into the portal blood, the amount of bile acid in the portal blood causes the body to respond in a certain way. So we have high levels of bile acid, and what would happen? There would be decrease in bile acid synthesis, right? So when I say we have decrease in bile acid synthesis, what does that mean? There's a certain step in the biosynthesis of bile acids that would get decreased. What is that? Yes, 7-alpha hydroxylation. Yes, this lessens. And then we also have increase in bile acid secretion. So we know bile acid, when it's made in the hepatocytes, it gets secreted into the canaliculi, right? And there's just very important transport protein that secretes it, that causes it to get secreted. Now, what's this? Yeah, bile salt export protein. Okay. Now similarly, and when we have low bile acid in the portal blood, the opposite would happen. We'd have increased bile acid synthesis, which means we'd have increase in 7-alpha hydroxylation, and then we'd have decrease in bile acid secretion, so we'd have less activity of BSCP. Do you guys have any questions? Mhm. Okay, now let's look into how bile is clinically relevant. So In bile, we have many substances. We have bile acids, the most important ones. We have phosphatidylcholine and cholesterol. These three exist in a ratio that is 10 to 3 to 1. It's not important to remember this ratio, but all we need to know is that any deviation from this ratio can cause formation of gallstones. Okay. When I say deviation, it could be increase in cholesterol, it could be decrease in bile acid, it could be decrease in phospholipids, and all of it could mess up the ratio and cause stones to form. Okay. Now let's say we have high concentration of cholesterol. This forms something called lipogenic bile. It's basically normal bile, but with very high cholesterol. Okay. So as cholesterol keeps increasing, it saturates. And this is called supersaturation. Okay. And okay. Now once the cholesterol is saturated, when it increases a lot, it precipitates to form crystals. Okay. And this is called nucleation, precipitation of cholesterol to form crystals. Okay. These crystals eventually they aggregate and they form a microstone. A microstone, it increases in size, and as it grows and increases in size, it causes gallstone. Okay. The formation of gallstone. Okay. So again, just to repeat it really quickly, we have lipogenic bile that's caused by that's basically increase in cholesterol, and then this saturates, forms, this is called supersaturation, and then cholesterol precipitates, that's called nucleation. Then they combine together to form microstones, and these they increase in size and they form gallstones. Okay. Okay, now these gallstones are different parts of the ducts that they can block. Now starting with the cystic duct, if the gallstone here blocks the cystic duct, now you won't have bile, the gallbladder won't release bile into the common bile duct, right? Does that mean we'd have no secretion of bile salts? What do you guys think? Yeah, no, because we still have the hepatic duct that would release its bile into the common bile duct. Okay, that will eventually get secreted. Okay. Now, would it affect this blockage, the cystic duct, would it affect the release of pancreatic enzymes? No, it won't. Okay. Okay. Now the blockage of this gallstone, the cystic duct, it causes bile to accumulate in the gallbladder. Okay. So when that happens, it eventually leads to inflammation, and that's called cholecystitis. That's one of the consequences of the cystic duct getting blocked by gallstones. Okay. Now, when the gallstones block the common bile duct, now what do you guys think? Would it affect the bile salt secretion? Yes. Now, why would it do that? Because we have both the cystic duct and the hepatic duct. They're both bile coming from both sides are blocked. Okay. Now, would it affect pancreatic enzyme secretion? No. Okay. Okay. So now the gallstone blocking the bile, it would do three things. One, again, it would cause cholecystitis because we have bile accumulating back into the gallbladder. And another thing, because we don't have bile salts getting released, we know that we need bile to hydrolyze lipids and to reabsorb it, right? So if you don't have bile getting secreted, lipids won't get absorbed. Won't get absorbed into the intestine. So if they don't get absorbed, they would get excreted in feces, and this is called steatorrhea. Another thing it does is because again bile doesn't get released, so it starts accumulating here in the ducts. And when bile accumulates, we know that bile is made of multiple components. One of them is bilirubin. Okay, so when it gets accumulated, it causes something called obstructive jaundice. Okay. Now when we have gallstones obstructing the sphincter of Odai, would you have bile salts getting secreted? What do you think? No. Would we have pancreatic enzymes being secreted? No. Okay, so it affects both bile cells and pancreatic enzyme secretion. Now again, because it's blocking, bile accumulates, causes inflammation of the gallbladder, cholecystitis. Lipid does not get absorbed, it gets excreted in the feces, that's called steatorrhea. And then again, bilirubin accumulates, that causes obstructive jaundice. And then because we don't have pancreatic enzymes getting released, they kind of accumulate there in the pancreatic ducts, and they start eating away the pancreas, and this leads to inflammation, and that's called pancreatitis. Okay. Thank you, guys. Do you have any questions? Okay. These are my contact details if you guys want to ask any questions. And please scan this QR code and fill the form for feedback.